So I will just ask Matt to really start the talk, and we are actually looking forward to his talk. Matt, can you get started?
So as Partha mentioned, I'm a postdoctoral scientist at the Marine Biological Laboratory. I've done a lot of work in super resolution microscopy, and particularly with FPOM back at the University of Maine with Dr. Sam Hess. That was my graduate studies. So I've done quite a bit of work in that field. What you're seeing here, this first image, is a super resolved NIH3T3 cell with a viral protein called hemagglutinin in green, and in purple, this cellular marker that marks PIP2, a lipid. So we were looking at connections between viral proteins and cellular components. But what I want to talk about today is, let's see, can I get this to go? There we go. Some of the concepts of single molecule localization microscopy, in particular, how it applies to FPOM, POM, STORM, and DSTORM. And then I'm going to talk about a processing workflow for these microscopy methods. In particular, what you should look for during data acquisition. And then I'll take you through a workflow that I use to pre-process the images, to detect point spread functions, localize and fit them, post-process the images, and then finally to render them. And towards the end of the talk, I'm going to cut out of the lecture here a bit, or out of the PowerPoint, and use some real data and show you how you might use some ImageJ or Fiji plugins to analyze some of this data yourself. It's fantastic if you know how to program and you can do a lot of these image processing things on your own, but of course not everybody can program and not everybody wants to program. It's a very useful skill, but there are a lot of pre-built software packages that are free, open source, that you can use to analyze single molecule data. So I'll show you how those work, and some things to keep out for, but I'll talk more about that later. So to start at the very beginning, we have to talk a little bit about the diffraction of light. Even if you have a perfect optical system, if you have a point source, you never see it as a point source. Instead, what happens is that point source gets blurred by the optics of the system. It gets convolved with the point spread function of the system, and that limits the resolution of your system, or how easily you can see two small things apart. So for example, in this case, these are 2 point spread functions far enough apart to be resolved at the top, and the right, you can see the just a line profile through it. And in the middle, you can see what's known as the Rayleigh criterion, which is when they're considered to be just far enough apart to be resolved. And in fact, there's another limit called the Sparrow limit that's a little bit closer. You can see the two humps are not quite touching each other yet. And so you can kind of resolve them a little bit closer together than the Rayleigh criterion, but because of other reasons like noise and imperfections in the system, usually the Rayleigh criterion is considered the minimal resolvable distance. And of course, at the bottom, when the two point spread functions have come together, you can't really tell them apart at all. And those are considered to be unresolvable. So to sort of overcome this, we have to talk a little bit about fluorescent proteins. And these methods make use of either photoactivatable fluorescent proteins, photoswitchable fluorescent proteins, or in the case of DSTORM, STORM, they're organic dyes that are combined with a chemical mixture to let them blink. But for POM, FPOM, let's just look at these photoactivatable and photoswitchable proteins. So you can tag whatever you want in the cell with these fluorescent proteins, or proteins in the cell with these fluorescent proteins. And in the photoactivatable case, the protein starts in a dark state. In the photoswitchable case, you can see the protein, but it's going to change colors. So when you hit it with 4 or 5 nanometer laser or close to UV, the inactive one can become activated, and then you can visualize it with another laser. In the photoswitchable case, it just changes color. So you're able to separate those proteins out using a dichroic filter or something like that. And if you hit them hard enough or for long enough, these will eventually bleach and then you won't see them again. So these are sort of essential to POM, FPOM, and the way that that works. So to describe how FPOM works, or STORM, consider some real pattern up here on the upper right. And if you blur it with the point squared function of the system, what you would actually see if you look through a microscope is what's on the right here, this blurry thing where you can't tell any of the details apart, since those are much too small to resolve. All of those point squared functions are stacked on top of each other. They're all on at the same time. And so you get this blurry mess. So to overcome that, we switch some of them on, localize them. So we switch them on, we find them, localize them, and then you can add them to a render. So you can do this over and over and over again. And because only a few of those molecules are on at the same time, you can resolve them. You can separate them and tell them apart. So you sort of overcome this diffraction limit by using this process. And as you can see on the render on the right, this is just a simulation, but the details of the original pattern become much clearer than if you were looking at the diffraction limited image. So you're just going to keep repeating this. And in some cases, you get unlucky, like in frame 100 here, you see the red boxes, where you have 2 point squared functions that are not necessarily resolvable. They're on at the same time. And so you wouldn't be able to tell those apart. But these methods are stochastic and they work based on localizing enough molecules to be able to tell the structure apart. So you don't need to absolutely get every single molecule that's there. You just need to get enough to sample the structure. And a typical setup is actually pretty simple. And this diagram might look a little complicated, but essentially you just have 2 lasers coming in towards your sample. They get focused to your sample, they get focused to the back focal plane of your objective and come out as a column if you wanted a wide field illumination. That excites your sample. Fluorescence from the sample travels back through a dichroic mirror and then just gets sent through a tube lens to a camera. So it's a fairly simple system. You can set it up in an afternoon if you understand these simple optics, and it's quite powerful. Now, of course, there are variations of this system. There's all kinds. You could set this up in a total internal reflection mode where you get just a thin layer near your sample, or you can use a sheet, or you can do all kinds of things, but this is just to give you an idea. So as an example, I'm going to start with some simulated data, and later in the talk, I'll use some real data so you can see some of the challenges with real data, but I want you to see how these things get corrupted by various forms of noise and electronics and optics and other things like that. So we'll start with kind of a perfect case, and then I'll make it worse and worse and worse, and then we'll see what we can do with it. So for our example case, I've just generated a bunch of airy patterns, which the equation you can see down here, it's a Bessel function. It's a little messy, but you essentially get this big central lobe, and then these rings around it. And the rings you typically don't see unless the molecule is really, really bright. And if it's really, really bright, you might see them, but generally you don't see them. So I'll start by simulating a bunch of these. So let's say we have a bunch of airy patterns plotted at random. They look pretty nice. They're pretty clear. We can tell most of these apart. Not all of them, but most of them. And I'm going to corrupt that with Poisson noise to simulate photon counting statistics. So when a fluorescent molecule gets excited, it emits a photon in a random direction, and you can't always, it's not always going to be a smooth, like nice, clean point spread function that you see. You'll see them corrupted like this due to the effects of photon counting. Camera read noise also comes into play here a little bit, also a Poisson process usually, but that's how it's corrupted. And then I'll add a little bit of extra noise to simulate some, for example, an EMCCD behavior where you sometimes get some hot pixels and other small corruptions in your data. So starting with this, how do we find all these point spread functions, localize them, and make a map of where all of these molecules were. Let me switch. And I want to mention briefly about how you should acquire the data, what your frames should look like if you're in the lab and shooting your sample with some lasers and catching some molecules on a camera. And there are three conditions I want to show you. One is just the sort of low density condition, where you have just a few molecules per frame and they're pretty easily separated. Not terrible, but it will take a long time to acquire images for your sample. I mean, if I tried to take it like this and I had millions of molecules, it would take me a very long time or a lot of frames in order to sufficiently reconstruct my sample. And it's also prone to drift because of that time. So that's something to keep an eye out for. If I have what I would call high density, much shorter acquisition time, you're going to get lots more per frame. But sometimes the PSFs overlap. And remember, it's not such a big deal if I miss a few molecules, because I just need to sample enough so that I can see my structure. If I sample 90% of my molecules, I can still see my structure really well. If I get even 66%, I can probably still see my structure pretty well. And then, of course, there's the extremely high density where I can't resolve most of these molecules. I definitely can't tell them apart. And I'm starting to approach the diffraction limited case again, where all of the molecules are on and they're all overlapping and I can't see any better than just with a regular microscope. So this sort of situation should be avoided. And you can always adjust your laser powers, camera frame rates, things like that, in order to get it to look more like this if possible, or even more like this if you don't need that many molecules. But this is sort of the ideal. So let's say that we have our data. We have some nice looking frames like this. Hopefully it looks something like this. It probably won't. And again, I'll show you some real data later to see what that looks like. But in my simulated case, it's pretty nice. What do we have to do next in order to localize all of these molecules, tell them apart, and get some final reconstruction? So to localize the PSFs, to localize the molecules, the first thing we have to do is detect them. And I like to pre-process the images in order to make that easier. And this workflow that I'm showing you, or from this point on, this workflow is sort of, I guess I'll say it's my preferred way to look at these, but it is certainly not the only way to process this data. So if you see other people doing different things, it's not necessarily that they're doing it wrong, but they're doing it a little differently than I am. But again, I've looked at a lot of data and this seems to work pretty well for me. And you'll see some of these other packages use a similar approach. So I think it's pretty robust. So the first thing I'll do is I'm going to take the raw images and I'm going to filter them to form an auxiliary image. And I'm going to use that auxiliary image to reduce the number of bad fits that I might get, and make finding the PSFs much easier. So starting from this raw data, the first thing I'm going to do is median filter it. And this might seem like an odd choice because it blurs the image, but this is just the auxiliary image. It's just going to help me later. So I don't care if I really blur this too much. I'll deal with that later. But the reason I blur it is because I get rid of those hot pixels. So you can see, let me see, is there a way I can annotate? Well, there's some hot pixels in the bottom right corner. You might see those little white dots. And when you median filter it, you're essentially taking a box around each of these pixels and you're replacing it with the median of those pixels. And if you have a really hot pixel or a really low pixel, you have any sort of outliers, it basically removes all of those. So I start by median filtering. And the next thing I'm going to do is apply this technique called the difference of Gaussians. And I do that because it's simple, it's very fast. Essentially what this is I blur the image with a Gaussian, and I do it with two different Gaussian radiuses, one that's, or radii, one that's small and one that's large. So the small Gaussian radius gives me an image that looks like this. And when I blur it with a large one, I get something that looks like this. And if you take the difference of those, so I take one and subtract the other, I get something that looks more like this. So what that does is it enhances the features of this image, because now these are much more visible. I mean, from the original image, not terrible, but now they're super obvious. They're incredibly obvious where the point state functions are. And what I can do is I can just threshold that and pick out my point state function. So I get this mask of positions. I can take each of those and just find the centroid of it, and that's sort of my starting place for where all these PSFs are. And what I'm going to do is take each of those and I'm going to grab out a little box around it that I'm going to use for fitting later. So this is just an example of a bunch of these are a bunch of grabs that I took from the original image. So the auxiliary image just helps me find where all of those are. And then from those locations, I pull from the original image, not the auxiliary image. So whatever processing I've done to the auxiliary image shouldn't make it into the grab here. All right. So now I've grabbed a bunch of molecules or PSFs from molecules, I think. What's the next thing to do? Well, we would like to fit those molecules to find their molecular positions, their coordinates, much better than the diffraction limit. The first kind of quick pass is to just look at the centroid, which is the center of mass of those objects, as if the intensities were mass. And I'll just give you a 1D example of a centroid. And so if I have a PSF here and it's been corrupted by Poisson noise, and if I look at the centroid, this actual molecule is centered around 0, and my centroid gets me pretty close to 0. And if I shift it over to 2, the centroid does a pretty good job there as well. It also gets me to 2. And if I add some arbitrary background to it, because we want our fit to be robust and work even if there's background or some other non-ideal situations, it still works here. But you'll notice when I shift it back to 2, excuse me, this arbitrary background pulls my centroid off target. And so centroids are really fast to calculate, but they're terrible for accuracy. So you definitely don't want to use them or publish results using centroid fits. They're really not that great. They're just sort of a first guess or a first approximation. What we want to do instead is try to find something that can fit our actual point state functions. And most of the time, our point state functions are in the form of an airy disk. But fitting this airy disk is actually a little tricky. It's a little messy, and these extra rings here in the corner don't really contribute very much. So what typically happens instead is we fit to a Gaussian, which is a very good approximation to an airy disk. You can see here the blue is the Gaussian overlaid on top of the airy disk. And it's close enough that in general, you don't need to use the airy disk. There are a couple of situations perhaps where you might want to use an airy disk, but it's really not used that much. And the Gaussian is a much simpler function to fit to, and we know the parameters, or we can find the parameters of the Gaussian pretty quickly and pretty easily. So what I'm going to do is then fit it to a Gaussian, and usually that's done using the sum of least squares. It doesn't have to be, but that's sort of the standard. There's also maximum likelihood fits that work quite well, but they're very similar in accuracy for the most part. So my Gaussian model is this right here, and in one dimension it looks like this. I have my maximum intensity is I naught, e to the minus x, which is my coordinate, minus mu, which is the center of my Gaussian, squared over 2 sigma squared, sigma being a parameter that tells you about the width of the Gaussian, and B is just going to handle my background counts. So B is the background, I naught is that maxima, sigma is the standard deviation, or in this case, it's going to tell us something about the width, and mu is the coordinate. So for the most part, we're interested in finding mu, right? We want to be able to localize these point squared functions and find their coordinate. But the other parameters are going to help out later. So we really need all of those in order to do a good job. Excuse me. And the idea here is just to minimize the sum of squared errors. And there's a lot of ways to do that. A lot of algorithms that already exist that do that. And we can end up with a localization precision that's based roughly around these parameters here in the bottom left. So this isn't quite right. I believe it's been updated. I think you have to add about 20% to this. This is a little bit too ideal, but essentially the localization precision, or the ability to localize the center of these point squared functions, depend on these parameters here. So the width of the PSF that you're using, or the PSF that your microscope has, the pixel size, the number of photons that you detect, and the background noise. And if you pick the right pixel size and you have low background, this sort of simplifies to r squared over n. So it scales with the number of photons. So more photons, better fitting in general, and lower background, better fitting. So you really want a bright molecule with low background in order to get the best possible accuracy. So to localize them, basically I take those graphs from the original image and fit a two-dimensional Gaussian to it. And you can see some examples here, the raw PSF on the left and some fits on the right as the algorithm is fitting them. It's only showing you a couple of iterations. It actually iterates, about 100 times, sometimes less, depending on what the tolerance is. But okay. So after fitting, what we end up with is basically just a list of data, a big table of fitting parameters. In this case, A is our I naught. Mu x and mu y are the x and y coordinate, or sorry, the x and y coordinates. Sx and sy are the sigmas in x and y, and B is the background. So it's important here to note that not all of the fits that you get are going to be correct. The fitting algorithm will do its best, and it will sometimes, fit very, very well to things that are not very physical. So for example, if I had a hot pixel that somehow made it through this algorithm and it fit it, it might give me a Gaussian with a sigma that's less than a pixel. But I know that my PSF must be more than that. So not all of these fits are possible, even if the fit is really good. So not all of these things are point squared functions. So we need to be able to filter these out before we analyze any of the data. We're going to end up with a lot of artifacts. And you can see some of these artifacts in the render I'll show you in the next slide. But for example, we can look at the sigma for Gaussian at 550 nanometers if you're using an objective NA of 1.4. And we would expect the radius of the airy disk to be around 240 nanometers and the Gaussian sigma to be around 83 nanometers. So if you got something really far from that, you don't want to include it because that's really, really far from the expected value for that radius. And you're probably fitting something that isn't a point squared function, or maybe you're fitting several point squared functions in one big clump. But in any case, you wouldn't want to use that in your further analysis. So these need to be filtered out. So for example, if I took this data set here and I plot my sigma, this is just a histogram of the sigma value for the Gaussian, so the radius of the Gaussian, I might get something that looks like this. And you can see this false peak on the left. And that probably is due to hot pixels or other bits of noise that the algorithm picked up and then fit, rather than actual point sort functions. And the ones that are really long out here towards the right are either out of focus or possibly multiple molecules together in a clump that all turned on at the same time. And so we want to take our expected and select some region around that in order to get rid of these sort of bad fits or non-physical fits, I should say, because they could be really good fits, but they might not be physically reasonable. And just to give you an example of what that could look like here, I have an image. This is a Dendra 2 hemagglutinin, and this is just a pretty dense set of molecules here. But you can see on the left one, there's this sort of gridding pattern that appears when I haven't filtered any of the bad pixels out, and so you end up with this strange looking artifact here. And so you wouldn't want to do your data analysis on any of that. And on the right, after filtering some of the parameters out, you get a much cleaner looking image free of these strange artifacts. I also want to mention something about resolution that often gets overlooked. And it seems like a very simple thing, but the resolution that you get from your imaging in this case depends not only on the precision of each of your localizations, but how many you got. So, you know, in the extreme case, if I had one molecule and I localized it really, really well, maybe down to, I don't know, 10 nanometers or something, but my structure spans the entire cell or my entire sample, I have localized my molecule really well, but my resolution is terrible because I haven't sampled that structure enough. And you can see in the example here, like if I look at just the first 1,000 frames, it's really hard to tell what this structure looks like. And by 4,000 frames, I'm starting to see what that structure looks like. I have many more molecules. And by 8,000 frames, finally, you can start to see that there's some structure here that that appears in the cell. So the resolution of the image on the right is much better than the resolution of the image on the left, even though possibly the localization precision of the left one might be better. There simply aren't enough molecules to be able to resolve the structure that I'm interested in. So that's seems simple, but a lot of people make these sort of mistakes. It depends both on sampling and on localization precision. So at this point, I want to take some of the time that I have left and talk about some of these plugins that are available for free. They're open source. I think they're all open source. Yes, they are. And they're available through ImageJ or Fiji. And if you don't have ImageJ or Fiji, I do highly recommend downloading ImageJ or Fiji. It's sort of a Swiss Army knife of imaging tools, and it's very, very useful. So I'm going to switch over. Oh, sorry. The three I'm going to talk about is QuickPalm, Thunderstorm, and this thing called NanoJ Squirrel, which is very interesting. So let me see if I can switch over to desktop. Hopefully you can still see my screen.
Yes, it's visible.
Okay, great. Let me get these windows out of the way. Okay. So Fiji is just this version of ImageJ that comes prepackaged with a bunch of plugins. If you get Fiji, it comes with Thunderstorm and QuickPalm, as well as a ton of other plugins that are of use. So we'll just load this up, and I have a sample data set. So I'll show you some real data that I took some time ago. And as you can already see, it's not quite as nice as my simulated data, but sometimes that's what real data looks like. And there's lots that we can still do with something like this. So this is taken with an EMCCD at 120x 1.45, and this is a Dendra 2 HA molecule that's been transfected into a mouse fibroblast cell. And I can just play through it a little bit so you can see each frame of this. There should be different molecules visible at different times, ideally. I can run it through. And you also see as we get towards the end, I start to bleach out a lot of these molecules, and there's some weird haze here on the left that shows up, probably from autofluorescence from the cell. And by the very end, we've got very few molecules per frame. And that's something that happens, of course, while you're imaging. As you start bleaching out molecules, the number of molecules per frame should reduce, and you can try to overcome that by increasing laser powers as you image, ramping it up, but eventually you're going to run out of molecules. So we have our data set, and we'll start with QuickPalm. And QuickPalm is in the Analyze menu here. And if you have Fiji, it's just right here at the bottom. So Analyze QuickPalm, and you can go to Analyze Particles. And unfortunately, there we go. Zoom is covering some of my options, but basically you have a bunch of options to localize these molecules, and I'm just going to use these settings here. So you can choose your signal to noise ratio. You can what your expected full width half max in pixels should be, and the pixel size of your data, which is something that you'll need to measure, or you can approximate it by calculating using the magnification and the pixel size of your camera, but you should really measure it if you want to be accurate. And I'm just going to hit OK and see what it does. And it will show me as it reconstructs. And it's going to go through all these frames. Okay. So with QuickPalm, it gave me something that looks like this. It's not the most pretty thing in the world. It just sort of binned these all together and tried to show me what the localizations would look like. And it gives me a list of all of the XY coordinates of these things over here in this window. And it's important to note that QuickPalm is not very accurate. It uses centroid fitting. And as I mentioned before, centroids are not very accurate. However, it just did 5,000 frames in, I don't know, a couple of seconds. So it's pretty fast, and this is great if you're in the lab and you're taking some data and you're not sure if you're really getting anything useful. But it's not especially accurate. So this is just to be used for sort of a first pass. I would never use QuickPalm or centroid fitting at all for any sort of data that you actually want to analyze further. But this is just sort of a quick way to get a look at what all of these molecules might show you. So that's all I'm going to do with QuickPalm, because it's not a very complicated system. And again, you probably don't want to use it for anything more than a first pass or a guess, or if you're in the lab and taking data. So I'm going to move on to Thunderstorm. And we're going to go straight to run analysis. And the first thing you want to do is, and in fact, it makes this very easy for you. It puts these all in order. Go to camera setup. And the first thing you want to do is choose your pixel size, the photoelectrons per analog to digital converter count, the base level, and your EM gain. If you're using an EMCCD. If you're using an SCMOS, I don't think this has options for that, but you can sort of get away with just turning the EM gain off. And the first thing here after setting your camera is the image filtering. So this is to generate the auxiliary image. So You have a bunch of different options for this. I like the difference of Gaussian's filter. You can choose your 2 sigmas, but there's a bunch of different options. You can do wavelets, averaging, Gaussian filters, those sorts of things. And you can play around with these settings to figure out what works best for your data. And they also have these little question marks here. Oh, if it doesn't break. Let's try it again. There we go. And it'll tell you what each of these filters does. So this program is really good about that. And okay, so let's choose that. And to approximate the location, let's use a centroid. And for subpixel localization, so this is how it's going to actually get the coordinates of your molecules. The integrated Gaussian does just fine. Weighted least squares fitting also does just fine. And you can choose your initial sigma guess. So this is what's calculated from your PSF. So in this case, I'll assume it's 1. It doesn't have to be perfect because it's going to do a fit as long as you aren't too far apart or too far away from the right answer. It won't take too long. And you can choose how it's going to visualize it. And again, you have a bunch of different options. I like the normalized Gaussian option. And we'll see what that looks like. So the first thing you do is hit the preview button. And it's going to spit out my auxiliary image here. So this is after being filtered with the difference of Gaussians. And it will show me the first frame and see what it picked up. So each of these little red markers is a molecule that the algorithm has picked up. And you'll notice it's not doing a bad job. For example, pretty decent. And that, for example, is questionable. Those don't look like points for functions. So it does make mistakes, and that's fine because we're going to filter the data later. But you want to make sure you don't have too many mistakes or it's going to take ages to run. But still, I'd rather be a little, I'd rather have a few more false positives than missed molecules. So this is a decent set of parameters, but we can always change these and see. For example, if I did a sigma of 1 and 2, hit preview again, see if it makes any difference. Still looks pretty similar. But this is something that you really should just kind of play around with until you get something that looks like it's picking up most of your points for its functions. All right, if you're happy with that, then you can run it. And it should give me an update every so often. There we go. So it's going to render it as it analyzes it. I thought it would be a little faster, but I guess on my laptop, it's not that fast. So it might depend on the speed of your system, but you can already see it's generating this sort of gridded pattern here. So it's making mistakes, lots of mistakes, but we're going to fix that in a moment. Still, it is making this sort of clumpy distribution, which is what I'd expect for this particular protein. I think perhaps I should have chosen a shorter set of frames to use, but we'll just let it go. So the way it's rendering this right now is it's taking each of those molecules and it's putting a Gaussian on top of it. So you can make these clusters very visible. So my artifacting is starting to spread a little bit. And it looks absolutely terrible over here. But there's going to be options to fix this later. Now, when it's done, what it's going to output is a list of parameters for the fits, a big long list of all the molecule positions, the fit parameters, so their sigmas and their XY coordinates and backgrounds. And it's also going to output the raw data with all of those red boxes on top of it, so you can always look through your data and see what it's picking up as a molecule and what it's missing. It's almost done here. There we go. All right. So now I have this huge list of molecules. How many did it find? It found 642,655 molecules, and it's glitched out my render. There we go. And usually, oh, there it is. It will give me the raw data back with all of these plus signs to indicate where the molecules were. Now, if we go towards the end, you can see this junk here is probably where all this gridding pattern is coming from, because these are not point squared functions. This is just noise. And for whatever reason, the algorithm has picked this up and thinks that it's point squared function. And so all of that is junk. So how do we get rid of the junk? What I'm going to do is go over here to my molecule list. And this has all the different parameters. Like I said, the XY coordinates, the sigma of the fit, the intensity, the offset, the background, and fit uncertainty. And I can sort by any of these. So for example, if I sort by the sigma, I have some crazy small sigmas here, like 0.006 nanometers, which is just not physically plausible. And if I sort the other way, I have some gigantic ones, like this 1.5 micron wide sigma, which is also not reasonable. So how do I get rid of those things? So you go down to filter here, and you can say, you can type, for example, sigma, and let's say we want only the sigmas that are greater than, I don't know, let's say 70 nanometers, and hit apply. And so it's going to re-render this. Any moment now. It's a lot of molecules here, so it's a little slow. Okay, it's re-rendered. And just by removing those really small sigma fits, I've pretty much removed all of that gridding artifacting that existed. And you can also add filters to this. You can say, okay, maybe sigma is greater than 70 nanometers, or is this little dashed line here, or sigma let's see, less than, let's say 200 nanometers. Oops. I think I want an and there instead of an or. There we go. And you can see in the bottom left-hand corner here, it tells you how many molecules were filtered out of how many you had. There we go. And you can repeat this process to kind of filter out this image and get rid of the really bad fits. And you can just keep adding to this list. I mean, if I wanted to look at just the molecules that have, you know, pretty good uncertainties, I could look at uncertainty, oops, and uncertainty less than, let's say, 100. And when you've got this image, so you've removed all of these gridding artifacts and you have the uncertainty that you want and the parameters that you want, you can export as a CSV, and you can open that in another program to do further analysis. That's where something like programming becomes really, really helpful, because you can then process these big lists of molecules and do other things with it, like cluster analysis, for example, or pair correlations or radial distribution functions. There's all sorts of analyses that you can then run on this. So there's one more thing I want to show you before I run out of time here, real quick, if I can. And that's this other plugin called NanoJ Squirrel. And I'm going to reopen this so I can show you. So what NanoJ Squirrel does is it takes this rendered image that you have, this super resolved rendered image, and it takes a diffraction limited image, which you can form by just summing all of the frames of your raw data. And it's going to compare the two using something they call it's the basically resolution. It's a Pearson coefficient that's been adjusted. So the resolution adjusted Pearson coefficient, I think that's what they call it. And it will show you areas in your sample that might be problematic, that you might have undersampled, or you might not have accurate data for. So I'm going to take this raw stack and I'm going to Z project it as an average intensity. So basically it's going to take all of these frames and just add them up together. And I'm going to get an image that is as if all of these molecules were on all at the same time. So I can always go from my PSF images to what this would look like, or my diffraction limited image. So basically on the right here, this is my diffraction limited image, and on the left is my super resolved image. If you ever want to compare the information that you can get from super resolution, this makes it really obvious. For example, these clusters are basically not visible in any of this, the data in the diffraction limited case. Okay, but let's see what NanoJ Squirrel says about this data. So I'm going to go to NanoJ Squirrel and run it. And I choose my two images. So I want my normalized Gaussian, which is my super resolved reconstruction. That's this image here. And I'll take my average stack. Oops. And run it. Okay. And it throws out quite a lot of data at you, but probably the most important or the easiest to interpret here is looking at this image here, which is our resolution scaled error map. So what this is telling me, these green regions here, it's telling me that I probably didn't sample that green region very well. In other words, there's a lot of difference, or there's a lot of error between my super resolved image and my diffraction limited image. And I chose this particular data set for a reason, because you can see here in the super resolved case, you have this sort of big black hole with a couple of dots in the middle. And in the diffraction limited image, you have this big blob here. And that's where NanoJ Squirrel is telling me I might have a problem with this green yellow color coding here. And the reason I might have a problem here is because I probably did not sample this structure. What likely happened, and we could always go back to the raw data and see, what likely happened here is that too many molecules were on at once. In other words, this was probably a very deep, very dense cluster. And probably when I was imaging, too many point spread functions were all on top of each other, all overlapping, all in the same place. And I either had to filter them out to get rid of artifacting, or it just didn't pick them up as point spread functions. So there was nothing there to fit. So you can always use this nano-J squirrel approach, this resolution scaled error map to sort of cross-check and make sure that you're really sampling. So if I were to say analyze this section of the image, I would want to be really careful about making predictions, because I think that I'm probably not sampling this especially well. All right, and I think I will leave it at that. And if anyone has any questions, I'd be happy to try to answer them.
Hi, sir. I have a doubt. How do you calculate background for each and every molecule?
Background, yeah. So I didn't talk much about background. There's a lot of different ways to calculate background. The one that I prefer is something called a temporal median filter. You can't access it from Fiji as far as I'm aware, but essentially what you do is you take your raw stack. Do I still have it open? I don't. Let me reopen it so I can show you. And essentially what you do is for each pixel, you choose a window. So say, I choose a window of maybe 50 frames or something like that. And looking through those 50 frames, I'll take the median through time for each of those pixels and subtract it off from that pixel. So what that does is, if I have a structure like this thing down here that's just sort of on all the time and just sort of glowing and not doing much, that will be the median of that set of pixels, and so that will end up getting subtracted off. There are lots and lots of different ways to get rid of background. I think Fiji supports only a couple. You can do rolling ball, which is a background subtraction method that's been around for ages, and does okay, but there are more advanced background subtraction algorithms. And you don't always want to subtract background either. That's the other thing. If your background is really low, remember in the Gaussian fit, we have a background term. And so if the background is locally flat, you probably don't want to remove background, because you could introduce other artifacts by removing the background. But if you have very structured background, it's important to remove.
Okay, thank you.
Thank you, Matt. It was really great. And, thank you so much. Sorry for the trouble initially what we had with the system.
That's
And,
okay.
Yeah, and I think this is one of the most beautiful talks I have come across, because this talks a lot about the fundamentals of single molecules and superas. And these the the candidates who are here, they're going to see the same thing, single molecules in the lab. So I'm really happy that you really talked on this, huh? And this is a great insight into the super resolution microscopy. And I feel that this talk will be looked upon as the entry point for most of the super resolution, imaging, and by graduate students who really want to enter this field. And great demonstration with image J, and Fiji. This was really practical. That's what they're going to look at. So it's really great. Okay?
Good.
And your talk is going to be there on YouTube for future generations to look upon, and they can actually, whoever wants to get into the field is going to be a great thing. So, Matt, I'm really happy with this. I does have a couple of questions that I thought I will ask for that.
Sure.
For the rest of the researchers around who want to get into this. Okay, so do you have a tips for practical tips for reducing background or increasing the brightness of the single molecules as you whatever we say? So what are the practical tips on experimental tip?
Yeah, so background's a tricky one, because that's very sample dependent. If you want to get a little more complicated, one of the things you can do is interlace your activation laser with your readout laser. So instead of leaving the activation laser on all the time and the readout laser on all the time, you can switch between the two, because a lot of the autofluorescence is more in the bluer range of light. So like if I leave the 405 on, eventually the cell will just start to glow and it just sort of accumulates. So the lowest amount of 405 you can use, the lowest UV you can use, you can avoid some of that cell autofluorescence. And if you interlace them, there's been some success in reducing that, because you have the 405 on, then you read out and capture. So if you can sync that with an AOTF or something like that, you could use that to reduce background. But that's a problem for all of these is cellular background, especially cellular autofluorescence. So smallest 405 you can get away with would be my first suggestion. And then if you want to be really complicated, you can sync your camera with an AOTF and have it alternate between 405 and whatever your readout laser is.
Yeah, Sam, sorry, Matt, there is another thing that I wanted to ask. How do you, where do you place FPalm with other super resolution techniques like STED, SI as of today?
Oh, I've lost my video. Oh, there it is. Where would I place it? So there is no one system that's great at everything, and so it's very hard to rank them, I think, because they're all good at very different things. And it's oops, my video keeps dying. Oh, we'll switch to the I think they're useful for different things. I think they all have their strengths and weaknesses. It's very hard to place one above the other. STED can get some great resolution as well using the sort of point scanning approach. I haven't actually used STED very much. It can be very hard on your samples because the laser powers are quite high, but that's also somewhat sample dependent. But they're all very useful tools.
Right. Okay, Sam, I think if anyone has any question, they can ask. Yeah, we have a question actually.
Okay.
Yeah, can you please come here and ask? I mean, that would, I don't know how the speaker system is going on. So yeah, come here and you can ask him.
Thank you. It's a very nice talk. It's a very informative talk. Although I have a question, like actually I was working with some blinking quantum dots, and somehow that get aggregated. Like, and if we take like 2000 or 5000 frames, but then if in each frame there remains one guy who is very intense, and the other blinking dots are too less intense to be detected, then what can be done in such types in this type of cases?
Oh, when they aggregate. So when they aggregate, if they're really, really close together, you have to worry about fret also, because you can have energy transfer between the dots. But you can't tell them apart, basically, is what you're saying. So they're basically all bright, or just a few of them are bright.
There are just a few clusters which are those are having too intense, but then the other ones, yeah, but then the other ones blurred due to these too intense dots.
Mm-hmm. Yeah, that's a difficult situation, because if all of the Poincare functions are on, or sorry, if all the molecules are on at the same time, you obviously can't tell them apart. And so there isn't a whole lot you can do from this perspective to tell those apart. You really need them to be separated somehow. It's an interesting problem. I don't have an answer for you, unfortunately. I don't have a good answer for you.
So one more thing is like,
Sure.
how much can we get into the depth? Like, how much can we get into the z-axis? Like, if we try to image it in the 3D format, is it possible using this type of systems?
Yes, so you can image in 3D. There's a couple of really good ways to do that. One is with called astigmatism palm, where you add a cylindrical lens into your system, and the point spread function above and below your focal plane will become stretched. And since it's asymmetric, it will stretch in a different direction above and below the focal plane. How thick you can go, probably not that thick, because what's going to happen eventually is there's going to be too much aberration for you to really measure those accurately. So I think, wow, I mean, I don't think I've gone more than, say, 10, 20 microns with that system. And I think with really thick tissue, you'd have a bigger issue. Yeah. But it is possible to get 3D information, but it works best, I think, on thin systems, thin cells or projections, things like that. Thicker tissue is tricky.
And the last question I'd like to know, like, if we are using these quantum dot type systems and they are blinking, so is there any limitation of their blinking time? Like, at least they have to be on for this time and off for this time, like that my camera can detect. Like, this will be the limitation with the camera, is it?
So some of it will be the camera, because the camera can only go so fast. If they're blinking really rapidly, as long as they're well separated, it doesn't matter. You'll just integrate them all into the camera frame and you can still tell them apart. But yeah. So some of that will be camera, but
okay. Thank you, sir. Thank you so much.
Sure.
I have a question.
Sure.
You have used photoactivatable fluorescent protein PAM-KATE, no? So how do you find a transfected cell which has this fluorescent protein?
PAM-KATE is very finicky. I show that as an example because I did use PAM-KATE, but it has a lot of pre-activation in my experience, and so when you first turn on your readout laser, there's just tons of PAM-KATE, and it doesn't respond very well to the activation laser, so it's hard to get more of it later in the set. It's not my favorite molecule, but it it does do its job. It's just usually those first few frames are just so bright that you have to kind of throw out some of the early data, which is really unfortunate. But that's been my experience with PAM-KATE.
So, like, how many frames will you be able to record using PAM-KATE compared with Dendra 2?
So, I mean, with Dendra 2, I can easily do 10 or 20,000 frames at 30 hertz. That's what I used to do. With PAM-KATE, probably the first 500 frames end up being kind of blown out, just because so many of them are on at once. So many are pre-activated.
Okay, is it true for all the photoactivatable proteins or only for PAM-KATE, like PAM-SERI, PAGF?
No, they all have different properties. So I mean, PIM cherry, for example, responds much better to 405 laser than PAM-KATE does, and is generally like more well behaved, but it's also a little bit dimmer. I've also used EOS, which is incredibly bright. I do recommend using EOS. It's very well behaved. It's very bright. Drampa. Basically, all of these have different quirks, and so depending on the combination that you're using, you might have to adjust your experiments a little bit to match. But yeah, PAM-KATE is a little different than the others in my experience, especially in regards to the pre-activation of PAM-KATE. There's just tons of it.
I have a question, sir.
Sure.
In Dendra 2, say what was the control you have used? Like Dendra, only Dendra, like before the control, there should not be a formation of cluster, right? in the control. So what was the control you have used for Dendra 2HA?
Oh, sorry. HA does cluster on its own. So, you know, HA is the fusion protein for the influenza virus, and it does form small clusters on the cell membrane. So
HA,
so that's
HA protein is like they form accumulation of the cells together, right? In the influenza virus, they accumulate the cells. Now, in the work you have done, you have said that the Dendra 2HA forms the clusters. Now,
yeah.
Dendra 2HA protein, there should not be any formation of clusters, right?
Sorry, I don't quite follow.
Dendra 2HA forms the cluster. This is what was the conclusion from the work which you have presented. Now, without Dendra 2HA, there should not be any formation of cluster, which means the molecules should be random.
If I just had Dendra 2, for example.
Yes.
Oh, yes. Yeah. That's right. If I just express Dendra 2 with no HA, it basically just ends up in the cytosol and it's everywhere. Yeah.
So in the work that is Dendra 2HA for influenza, that virus which you have reported, you have not put that control.
Sorry, the control that I showed you is not a control for Dendra 2. It's a control for mutation study that I didn't show.
Okay.
So that's yeah, that's I'm sorry about that confusion.
Okay.
So I guess, I mean, there will be questions, more and more questions. Is such an interesting thing, and I mean, this is not going to end, you know. So I finally thank Matthew Parent, Dr. Matthew Parent from Marine Biological Labs, Woods Hole, and thanks a lot, Matt, for coming down and, you know, spending your time. It's night now. I guess it's 12:30 or 1 o'clock. I mean, that's great of you that you could make it up. Okay, thanks a lot, Matt. Thanks a lot.
Yeah, thanks for having me.
Right.